going through this blue route where it avoids the complete oxidation of ammonia to nitrate, there's also a process called anamox, that's anaerobic ammonia oxidation, that combines, that uses ammonia and nitrite, and then directly remove it through nitrogen gas. And this way, it avoids all the nitrification processes through nitrate and then denitrification through nitric oxide, nitrous oxide, and all the way to nitrogen gas. However, these shortcut biological nitrogen removal processes rely on some of the very slow growing microorganisms. For example, the ammonia oxidizing bacteria, that's autotroph, and also anamox bacteria that has a really slow growth rate. And then there's this challenge of selective enrichment over other fast-growing microorganisms in the system. So there are some technologies right now that help us retain this specific organisms. And then here I have showed 3 examples. One of them is granulated activated sludge, granular activated sludge. So this rely on this self-organizing activity of microorganisms and forming this granular structure by secreting extracellular polymeric substances, or EPS. And then these allow them to aggregate and settle faster in the system. And then through this formation, they can have various zones with aerobic zones on the outer layer, and then anoxic or anaerobic zone at the core of the center. So of this granule, so that different microorganisms can be enriched all together in one system. And then similarly, there's also biofilm processes, MBBR, that relies on growth of microorganism on a media. So when you supply a media for them to attach to, they will stratify and then become thicker. As the layer becomes thicker, they will have diffusivity of oxygen from the outer surface, having a zone of aerobic growth near the surface, whereas near the media, they will have anoxic zone. And then again, during this process, you can enrich for ammonia oxidizing bacteria near the surface, and then anamox or other denitrifiers that leaves in the anoxic zone. And lastly, there is also membrane-based technology for biofilm systems, MABR, that relies on hollow fiber membrane to supply oxygen, or in this case, I'm showing MABR supplying methane through hollow fiber membrane. And then in this case, the diffusion direction is reversed, so that the oxygen or substrate gradient actually comes from the hollow fiber side, and then decreasing over this thickness of the biofilm that grows on the hollow fiber. And then in that case, you can have an aerobic zone near the surface of the membrane, and then anoxic zone where anamox or other denitrifier can grow near the bulk liquid side. Right. And there's also another way of enriching or retaining biomass, and this is something I'm going to talk about today, is the microbial encapsulation technology. So I would like to think of encapsulation as an engineered biofilm, because it shares a lot of similarity with biofilm systems, but we have much better control over this technology. So that I'd like to call it engineered biofilm. And then this is my metaphor here on how this encapsulation works. So on the left-hand side, I have a rope that we can tie it into a web with a lot of knots. And then when you have this web structure with a pore size that's small enough, you can retain different ocean microorganisms, right? Like fish and sea stars that's larger than the pore size. And then this is very similar to the encapsulation technology. In this case, I'm showing alginate as the encapsulant. So alginate has also this linear structure that's similar to a rope that has repeating units of sugars inside. And then in this case, when we add calcium to it, the calcium cation will be acting as nodes to bind this linear polymers together. In this case, the calcium binds to the negative charged network and form this eggshell structure that can form a gel-like material. And this process is called cross-linking. And after cross-linking, bacteria can be trapped inside because the pore structures formed is much smaller than the size of the bacteria. In that case, they're like those fishes captured in a fish net. At the same time, water can still go through these pores and then carrying substrate and electron acceptor, electron donors through this network so that they can to sustain the leaving of this encapsulated biomass. So with encapsulation, we can retain biomass similar to biofilm systems. At the same time, we can also develop this anoxic aerobic microenvironments inside each encapsulant. So here I'm showing a relatively old literature that's back to 1996, but they showed a really good diagram on how oxygen diffuses inside through. So this spherical structure is a encapsulant that has a lot of microbes inside. And then when oxygen diffuses from the surface into the core, you will see the gradient of oxygen going down that's consumed by the aerobic bacteria near the surface that converts ammonia to nitrite. And then you see a co-occurring decreasing of ammonia and the increasing in nitrite. And then in this region, you will be able to enrich for either anammox bacteria or denitrifiers that use nitrite as their electron acceptor. So that's the similarity between biomass, biofilm and encapsulated system. There are also some differences between biofilm systems and encapsulated system. For example, they are called engineered biofilm for a reason, right? So we can have a complete control over its initial inoculum. So unlike biofilm systems that develop over time, that rely on the self-formation of biofilm and the excretion of EPS by the bacteria, we can use encapsulant to trap whichever bacteria we want to start with. And then that should give us a much faster startup time for this kind of system. And then there's the encapsulant itself can also be tailored for different applications. For example, here on the right-hand side, I'm showing an SEM figure showing the structure of alginate that has a zeolite. The cubic particles are the zeolite particles that's being incorporated into alginate network so that this encapsulated material itself can have absorption capability to ammonia. And with that capability, it can help enrich ammonia metabolizing microbes, such as ammonia oxidizing bacteria or anammox bacteria. So this is just an example of how encapsulant can be tailored for different applications that's not limited to biological nitrogen removal. And so the encapsulant can also provide protection of encapsulated microbe. So here is a paper published by Dr. Novak's group back in 2020 they're showing the differences between a suspended culture of hydrogen-producing bacteria and encapsulated encapsulated culture of this from the same source. So that the black line is showing that the encapsulated microbes were not actually affected by their dosage of copper and dichromate. These are the known inhibitors for hydrogen-producing bacteria, microorganisms. And then with encapsulation, we're seeing a much harsher inhibition that's experienced by the suspended culture that's shown in red. So this way, encapsulating microbes can give them competitive advantage over suspended culture, and also allow to retain that selective inoculum you want to start with. And also note that this Y scale is actually on a log scale, so that's a very significant difference between encapsulated and suspended culture. So due to this advantages from encapsulation, it is widely used in environmental engineering. So here I showed 3 examples, including resource recovery, nutrient removal, and also remediation of contaminant. So on the resource recovery side, this is a review paper also published by the Novak group back in 2021. So they're showing the idea of encapsulating anaerobic microbes. So anaerobes are known for their slow growing and also sensitive to a lot of inhibitors and even oxygen, since they're anaerobes. And this make encapsulation technology very promising because they can protect them from this outer environment and also separate them out from the suspended culture. So with encapsulation, anaerobes can be used to recover resources from organic waste, such as industrial wastewater and municipal wastewater, and converting all this waste organic to valuable products, such as methane and hydrogen. At the same time, it will provide protection to them against the common inhibitors that exist in industrial wastewater. For example, heavy metals and other inhibitors that's commonly co-occurring with the organic waste substances. And then as for nutrient removal, this is another review paper that we published during the pandemic. This is building on my previous mentioned ideas of slow growing biological nitrogen removers processes, so such as Anamox and other autotrophs, AOB, AOA, and Comamox. So with encapsulation, we can retain them in a system and then achieve a single step nitrogen removal, so that when we supply oxygen in the bulk liquid, suspended culture will consist mostly of heterotrophs that grow on oxidation of organics, whereas when oxygen diffuses into the encapsulant, the outer layer will enrich for aerobes that oxidize ammonia, and the inner layer will consist of anaerobes, where they denitrify and remove nitrogen through nitrogen gas. So the last example here I have is on chemical remediation, so that we can also use encapsulant to trap reactive particles to remove pollutants in this way, utilizing the diffusion property of encapsulant. So these applications are also demonstrated at scale. Both hydrogen and methane production were demonstrated at pilot scale. In this case, this is also from Dr. Novak's group. showing a treatment of brewery wastewater. In this case, they demonstrated that encapsulated system has a faster startup rate than a membrane type reactor that's running in parallel. And this encapsulation reactor is used for both hydrogen and methane production, and it has showed low energy demand and capital costs. At the same time, it doesn't require membrane cleaning. On the other side, microbial encapsulation can also be used to upgrade existing centralized system, such as drinking water polishing for Micro-V from California. These are some of the commercialized solutions with microbial encapsulation. And Biocastle from Israel, you can use this cage-like structure to dip the encapsulants into existing infrastructure so that you can have an improved nutrient removal from the system. And also Hitachi from Japan is doing a similar configuration where you add encapsulant, it can be anammox or other bacteria into the conventional activated sludge system. So despite the wide application of this technology, there's still a lot of knowledge gap exists when we talk about encapsulation. So the biokinetics of encapsulated microbes still remain unclear. Some reports say there's increased activity. Some say there are decreased activity when microbes are encapsulated. And then we do need to separate out, sort out these reports from the effects of mass transfer as well, because that will also affect their biokinetics when they're encapsulated. So here, our review showed that there's a lot of microscopic processes that we may not currently still understand, such as the migration of microbes inside the encapsulant if it's flexible, and then if the encapsulant is not strong enough, there could be potential escape of microbes from the encapsulant, and then there's constant mass transfer from the bulk liquid with the encapsulant, with the microbes inside. And then as engineered biofilm systems, we can actually refer back to some of the knowledge we have on biofilm system. So the interactions between encapsulant and the encapsulated microbes needs to be explored as well. So other than the mass transfer processes such as concentration gradient and absorption, there could also be potential cooperation and competition between different microorganisms inside. So in this seminar, I hope to showcase some engineering approaches that we use to tackle this problem. So we used a bottom-up approach by establishing a model based on our understanding, and then build up a simple model system consisting of pure culture and alginate, so that we can use it to verify our model and then improve it. So this is an iterative process that we're going through with model quantification, verification, and validation with experimental design. And also, I'm going to show some of our recent progresses on developing a new method in validating the model results, the cross-section and fish method. So the first item we need to decide on when we establish a model is to determine the mass transfer property of the encapsulant we use. In this case, it's calcium alginate. So we simply use this diaphragm cell system to measure the diffusivity across alginate membrane. So we have two vials. When we add substances into the source vial, we track the change of concentration in the test vial over time to see how fast the substrate diffuses through this membrane made of alginate. And then we repeat that for all the key substrates in biological nitrogen removal. That includes ammonia, nitrite, nitrate, and oxygen. At the same time, we did experiment to measure the partition coefficient that's showing on the right-hand side. So a partition coefficient close to 1 means there's no preferential partition between the alginate phase that's on the y-axis and the aqueous phase on the x-axis. And in the case of ammonia, we actually observed a little bit of a slight absorption of ammonia onto alginate. That's shown in the blue line. So with this knowledge in hand, we actually built up a conceptual model that's shown on the left-hand side. So we added alginate phase to the one-dimensional biofilm model. So we modified based on a biofilm system, a model with the Aquasim platform. So we added a biofilm phase that can not only retain or absorb ammonia. In this case, the C ammonia alginate is referring to the absorption of ammonia on alginate, but also it can occupy some space that may be competing with the encapsulated biomass. That's the X alginate and XN. So with this modification, we are hoping to simulate the reaction between microbes that's being encapsulated and all the reactive chemical species, as well as with their environment, the encapsulant itself. With experimental design, we used a pretty simple system, calcium alginate as the encapsulant, and then a pure culture of Nitrosomonas europaea as the studied microorganism inside. And then this is to show the scale of this encapsulant. We made it with the diffusion preparation method. So they're about 2 millimeters in size, in diameter, and they're beads in structure. And then we studied both batch tests and also continuous flow bioreactor systems, so that all these experiments are done under non-sterile condition. So we would like to test out its capability of retaining this pure culture inside. So this is the result from the batch system, showing that the model actually predicted the growth of Nitrosomonas when they're encapsulated. So the figure A is showing its overlay of experimental results in dots, and then model predictions in lines. So we're seeing the consumption of ammonia in black, and then production of nitrite in blue. And on figure B, it's again overlay of experimental results from qPCR of AMOA genes that shows a growth over time. So this growth is actually resulting from the aerobic enrichment of AOB. And then since we extracted the DNA as a whole beads and then homogenized it, so you can look at it as the average of biomass concentration over the entire depth. So the red lines are actually model simulations at different depths inside of the beads. So at 0 is a darker red, and at the surface, which is 1 mm in the radius, that's at the surface. So over time, it's showing a preferential growth near the surface. And that is actually explained by the model with the following two figures, the C and D, with x-axis as time and y-axis as again as radius. So 0 is at the core of the bead and 1 is at the surface of the bead. So the inhibition actually potentially occur at the core over time due to oxygen limitation, both as a result of mass transfer and consumption by Nitrosomonas, and also a lower pH region developed near the core, figure D. So that's the result of ammonia oxidation process. So these results are published last year on Water Research. So with this tool in hand, we actually tested out several design parameters for encapsulated system. For example, the bead size, how big the radius, the diameter of the bead will affect the diffusion of oxygen. And also we tried different inoculation concentration to see where the which can be the optimized ratio of microbes and encapsulant. And also we tested out different aeration and nitrogen loading in continuous flow reactor that has a much more complex flow condition and also shear stress. So on the right-hand side, it's showing the overlay of simulation and experimental result in the continuous flow reactor. So we start to see some deviation after a long operation of the reactor, since we observed the breakage of alginate, since alginate is not strong enough to hold for a long time operation under non-sterile condition. And then also this is exacerbated by the higher aeration rate at higher dissolved oxygen points. And then the model simulation showing at the bottom here, the preferential growth near the surface, that's the light red line. And then the bottom figure is actually a zoom in of the top figure, where we have a much slower growth within the encapsulant, and then negligible suspended growth on the outer, that's the dark line near zero outside the encapsulant. So actually the growth near the surface is on the order of magnitude higher than encapsulated biomass. And then we verified this prediction through cross-section fish. So we cut the beads open, slice it into thin sections, and then stain it with fluorescence dye to highlight the ammonia oxidizing bacteria in red, in green. And then counter dye is the DAPI, stands for all the bacteria. So we start to see a very homogeneous distribution of bacteria at the beginning of the day zero. And then as it develops, as the reactor continues to operate, a ring structure develops near the surface. That's to show the preferential growth. And again, during long-term operation, the beads start to break up, especially under high aeration rates. So this is more of a qualitative analysis. So we want to see if there's any more information that we can extract out of this method. So we looked at some of the existing method tools for studying encapsulated system. So there's a lot of microscopic tools available to look at biofilm morphology, for example. There's optical microscope that looks at macroscopic structure, and then OCT and MR are really popular to look at mesoscopic structure. And until we will get down to like micrometer size, the SEM and the confocal lens microscopes can give us more information on the cell clusters and microscopic structures within the encapsulation. There's also microsensor tools that we can use to track the concentration gradient. So this is a publication from the Ishii group back in 2015, comparing granular active sludge and encapsulated anammox with their gradients in oxygen, in pH, ammonia, and nitrite. And then there's also a qPCR method that can track, that can quantify different nitrogen metabolizing microorganisms. So this is also from Ishii group, that they developed an assay to track all the nitrogen metabolizing bacteria, and then using different sets of qPCR probes. With a microfluidic system, we can get a high throughput qPCR information on that. So seems that there's no existing method that can provide us the information on a spatial and depth profile of microorganisms inside the encapsulant. We decided to modify the existing cross-section fish method to give it a to have a quantitative analysis out of the images. So we sliced the entire beads 100 micrometers apart, and then taking these thin slices, and then we add fluorescence labels to it to highlight different groups of microorganisms. so that we can differentiate them and have information on their size and colony count when they're encapsulated with the resolution of spatial distribution. So with this, we used actually a little bit more complicated system when we encapsulated Anammox enrichment, and again added a suspended growing Nitrosomonas pure culture. And then this is how it looks like when we encapsulated it. So you can still see some flocks of Anammox, and then Nitrosomonas is a suspended culture inside. And then we used batch reactors to incubate them by adding ammonia and nitrite. And then this is the performance showing over multiple cycles that we removed ammonia and nitrite at the same time. And then we collected samples from these beads, cross-sectioned them before and after the incubation for comparison. So we also did qPCR just to check if there's information that we can, as expected, qPCR didn't show any spatial information, and then they actually showed there's a decrease in concentration over time for all the probes that we used. Total bacteria for 16S, AOB for AMOA genes for AOB, and Anammox probes and NOB probes, all of them show decrease in concentration of cell. And this is actually over depth. So averaged over depth. It doesn't tell us any information on spatial distribution. So with microscope, we are actually able to see the colony develop over time. So this is a comparison of ammonia oxidized bacteria lit up by fluorescence dye from the before and after the incubation. So we see some colony developing. And with these images, we at the end we'll have a pile of images every 100 micrometer, and then we extract depth, count, and size information of the encapsulated colonies, and then plot it out. So this is the result that we have. So I can quickly walk you through. These are like 3 dimensions of the information. Basically, the x-axis is the depth for all this figure. From the surface, which is 0, all the way to core, which is 1,200. And then on the y-axis, we have colony size on the left, and then counts on the right. And the different time points are, the first row is initial, that's before incubation, and final after incubation. So we see that at the beginning, we have a more homogeneous distribution where this is showing the AOBs, the Nitrosomonas. So they have more homogeneous distribution over depth, both in both size and count, because that's what we used. We used a suspended culture that's more homogeneous. And over time, we see they have actually preferential growth near the surface. So there's a lot more count increasing near the surface. We also see some deviations between the triplicate that we use, that's denoted by the different colors. And then the colony size also increased near the surface as well as near the core. And then we repeated this as well for the Anammox. And again, the same layout of data. We see initially, actually, Anammox has a much more deviation than AOB because they started with granular sludge, and each bead can have a very different count of flocks that's being encapsulated, less homogeneous. However, over time, we see that the count starts to converge. So some of the excessive flocks die off, whereas the small ones start to grow. And then we see also a preferential growth near the surface, but it's not exactly at the surface. It's probably like 300 to 400 micrometers from the surface that's co-occurring with the AOB peak concentration near this depth. And colony size-wise, they actually shrink when you compare it to the initial colony size of Anammox. So in summary, in this seminar, I hope to give you some of these take-home messages. So we do think encapsulation is engineered biofilm. So existing biofilm models can be used to model encapsulated system with some modification. And that is also why we can use encapsulation to build model system to study biofilm behaviors, and allowing wider application in not only environmental engineering, but also the food industry and even medical applications. And then secondly, I showed just now the mass transfer properties of encapsulant can be very significant. It affects the growth of all the nitrogen removing bacteria inside. And then it also determines where and how fast they grow within the encapsulant. And lastly, with new tools that we can study and validate some of the model predictions, and then go through this iterative loop of simulation and validation to further understand and advance this technology. Moving forward, what we learned from our study showed that there's actually still a lot to explore. So there's still a lack of understanding of all the microscopic processes. For example, the microbe encapsulant interactions. So here on the right-hand side, I showed an SEM image showing bacteria colony growing near the surface can actually rupture the encapsulant if it's not strong enough. And that's actually what happened in our alginate reactors. And then these cracks can not only help them escape, but also change the mass transfer property of the encapsulant. And it also may allow invasion of suspended species from the bulk solution entering the encapsulant. And secondly, multi-omics approach can be used to understand the metabolism, whether there's altered metabolism of microorganisms when they're encapsulated. So with encapsulation, we can build cleaner systems with single species so that we can understand it with, for example, this transcription network analysis. And also with multiple species, we can study their competition or cooperation. And then incorporating all these processes into metabolic modeling can actually allow more predictive application of this technology in the environmental engineering field. And lastly, I would like to acknowledge some of my advisors involved in this in this project. So Dr. Paige Novak and Dr. Satoshi Ishii from University of Minnesota. They were my advisors during postdoc, which is most of these projects that I talked about today. And then our collaborators from the University of Notre Dame, Dr. Rob Nerberg, and he's a postdoc at that time, Patricia. These projects are funded by University of Minnesota as well as MinDrive through the biocatalysis initiatives. And this is my contact information. Thank you so much for your time.
Okay. Thanks, Dr. Wang, for giving us such an excellent report, which mainly referring to microbial encapsulation for enhanced nutrients removal. The microbial encapsulation is an emerging technology for removal of nutrients to remediation of contaminants. And in this report, Dr. Wang introduces us the challenges of biological nutrient removal and the advantages of microbial encapsulation. Also the differences between biofilms and encapsulation, and gives us 3 examples in the application of microbial encapsulation, including the resource recovery, nutrient removal, and remediation. And now it's time for the discussion. Is there anyone who wants to share your own opinion or ideas or questions with Dr. Wang? Okay, maybe I'll start because from last year, I am considering doing some research about the effects of biofilms on the transformation and migration of contaminants like fluoride arsenic in the groundwater. And of course, it is only at the start stage, because in your PPT, and I see you talks about the differences between biofilms and encapsulations. And I wonder, is there any different methods of characterization for the encapsulation and biofilms? Because for the start of your PPT, you state that the encapsulation also refers to the EPS, right? So I wanted to know if you measure the concentrations of the EPS or the live cells, dead cells, or the protein contents in the experiment.
Inside the encapsulated system?
Yeah.
So we didn't measure EPS in the encapsulated system, but we did carry out like live and dead stains of the cells so that we know which part, for example, so that can be used actually in combination with the microscopic tools so that we can see, for example, in this case, we can see which bacteria are alive or dead, because the DAPI will stain for all the DNA materials, no matter whether they're alive or dead. So that's why we used we tried out live dead stain to show the difference between those, but we didn't measure the EPS concentration inside encapsulated systems. So the other question you have is on analytical tools. So these are some of the examples I showed for analytical tools, which are also applicable to biofilm systems. So the morphology side, these are actually mostly used for biofilms these days, but I think they can also be used for encapsulated systems. I haven't seen much reports on them being used yet. But I think I do see a lot of similarities between the two processes, and I think these can be applicable and relevant. And then the microsensor side, there's a lot of use on both ends. So piercing through the encapsulant versus piercing through a biofilm can also tell you the concentration gradients of all that. Does that answer the question?
Okay. I also wonder if there is any differences for the microbial activities in the intracellular, cellular, and extracellular for the microbial encapsulation.
You mean encapsulated versus suspended?
Yes.
Okay. So yeah, actually that's a very good question. So I think that remains to me, I think that remains still mysterious because I do see a lot of reports saying that there's altered altered metabolism when bacteria are encapsulated. But again, as I mentioned, you do need to separate out the effect of mass transfer. That is the same within either biofilm system or encapsulated system, because when you have a gradient of diffusion, the activity you're measuring is not really the same when they're growing in suspension culture, right? And then I think that's why multi-omics approach can be helpful in understanding their metabolism when they're encapsulated. Another interesting fact is that, for example, bacteria growing inside encapsulant actually are standing a lot of pressure. So it's like you're trapped in a very small room with a growing population, right? And then the room has a limited space, and eventually it may explode if it's not strong enough, right? So bacteria growing under pressure can actually build up to a few PSI of pressure inside the encapsulant. And then under such pressure, I think there could be altered metabolism from encapsulated versus suspended free cultures.
And also, there is one scholar want to ask, is there any differences for the microbial encapsulation and microbial embedding?
Embedding as in, I'm not sure what this is referring to. To be more specific.
Maybe the embedding is more like the biofilm.
Yeah, I'm not very familiar with the term embedding because I'm not sure. Usually in biofilm, they refer to like immobilization. That means to fix the bacteria in place. That could be a biofilm system mostly. And then recently, I think a lot of people are using the term encapsulation for this kind of technology. That is to refer to using permeable matrices like alginate that are permeable to different substrate and then trap the bacteria inside. It doesn't have to be a shape in the shape of spheres. That's just how we prepare the beads. You can also be like coating of alginate on some substrate that can have all kinds of shape, right? So it doesn't not necessarily have to be a spherical structure if this is what the question is asking.
Okay. I still have another small question. Like in our groundwater system, when the minerals like clays, ferrum oxides could also remove contaminants by adsorption process, that is through the adsorption process. But in some appropriate environments, the adsorbed contaminants or the adsorbed fluoride, arsenic could be dissolved into the groundwater system. So I wonder, is there any consideration for the release of contaminants after the microbial encapsulation?
You mean at the end of use as in
yes.
like after using a disposal of these microbes? Oh yeah, that's actually a very interesting question. So I think it may so if you're concerning about like heavy metal and all that stuff, this encapsulation will be similar to say a waste sludge, right? And then even I think it also has some advantages if you're talking about like life cycle thinking of end of use of all this material after at its end of life when disposing them. Encapsulation actually I think personally, I think it has benefits over biofilm system that rely on substrates, right? If you put a lot of plastic supporting material to grow biofilm in treatment systems, and then at the end of use, end of lifetime, you still need to dispose those. And then these kind of substrates are actually plastic that degrades at a much slower rate than any encapsulant that you use. Usually they're biodegradable, like alginate and some other material that derived from the nature.
Okay, thank you. Also, there is Xiaoyang Tang wants to ask, do you think they have any application prospects in the removal of wastewater nitrogen and organic matter?
You mean its application?
Yes, application
yeah.
in the yeah.
So I've showed a lot of applications for organic waste removal and nitrogen removal. And these technologies are actually being commercialized right now. So there's a lot of company commercializing this technology for wastewater treatment. Yeah.
How about the removal of wastewater organic matter?
Oh, as in organic matter? So unless to my I think because this technology is more for retaining say slow growing or sensitive microbiomes. If it's just to remove like waste organic that can be easily degraded, I don't see a benefit of doing so because suspended culture that grows pretty fast on these substrates, unless it's a contaminant that's very hard to degrade and you need specific microbes that will fall into say, contaminant remediation category that I talked about in the 3 kind of applications.
Okay. Is there any more discussion? Maybe scholars are shy and they want to exchange ideas, maybe through email with you.
Sure, yeah, I have my contact information there.
Okay. That's all for today's report. Thanks, Dr. Wang, again for sharing the microbiome encapsulation. Have a good day for everyone. Thanks again.
All right, thank you. Bye.
Bye.